Hello again. Let's talk some about a variety of different cellular organelles. But in this case, I'm trying to focus on those organelles that are specialized for energy conversion, that form part of the structural support or movement components of the cell. And so it's sort of a mixed bag, but it covers a number of important organelle structures found in eukaryotic cells. First of all, let's consider the organelle called the chloroplast. This is an energy converter, and what we have here is we have an organelle that has enzyme systems, protein systems within it, that allows the organelle to convert solar energy into chemical energy. We'll talk more about this process later, as you might imagine, and the process itself is going to be associated with an event called photosynthesis. We find these chloroplasts in plants, and there's also some protists that have chloroplasts as well. What are we going to be doing with these chloroplasts? And the process is fascinating. In the photosynthetic process, we're converting solar energy, energy from the sun, into energy stored in organic molecules, sugars. So I know that you're going to look forward to, and it's going to be fascinating to see how these different portions of the chloroplasts are associated with this process of photosynthesis. Another organelle that's important for energy conversion is the mitochondria. Think of the mitochondria as being all sort of like a furnace, right? Sort of like an engine of a car. It takes one form of energy, chemical energy, and converts it into usable energy for the cell or the organism. So what's happening? The mitochondria carries out an event that we'll look at in detail, all the steps of it, called cellular respiration. And this is a process where we take a food molecule, some kind of resource molecule, an energy molecule, and we're going to break it down, and in doing so, produce a short-term energy storage molecule the cell needs called ATP. Here's what the mitochondria looks like. Notice the complex folded membrane structure like the chloroplast, and you might want to reflect back on the idea of endosymbiosis and think about how this mitochondrial organelle was believed to have been, at one time, a prokaryotic cell. So make sure you give yourself some time to think about that. Notice the complex folding and internal structure of the membrane. All of that will become important to us when we cover that process called cellular respiration. Now, inside of the cell, there's a support structure. Just like you have a skeleton inside of your body that provides structural support and is important for movement, also within the cell, there is a protein structure called the cytoskeleton. It's the cell's internal support system. Helps organize it, and also it's going to be related to some of the activities that the cell carries out. Made of protein fibers. These protein fibers, as you can see, are interwoven into a meshing network. It can be quite complex, and the important thing to think about is it can be reorganized. So the cell can take it apart and put it back together as the cell needs to change shape, or to change form, or to carry out new functions. So don't think of this as a static structure. Think of it as a dynamic structure that can be modified and changed. All right, so what do we have as the key components in the cytoskeleton? We have microfilaments, and the key one is a microfilament called actin. That's the name of the protein that's associated with these microfilaments, and we find actin associated with a number of movement activities carried out by the cell. So when the cell wants to change shape or move or change form, we might expect to find the microfilaments composed of actin to be involved. There's also larger filaments called intermediate filaments, and as you can see, we think of these as being protein structures that are associated with structural roles. Reinforce the cell structure, anchor the cell to another cell, maybe help to position or anchor organelles within the cell. So notice here we're talking about a structural role for these intermediate filaments. And then we're going to see structures called microtubules. And microtubules have a number of roles within the cell, but notice their name, microtubule, which implies that we're talking about a tube-like structure of protein. So microfilaments, intermediate filaments, and then the microtubules. They can help give or provide support for a cell. They can be one of the components of the cytoskeleton that anchors organelles. They're a key way in which materials get moved within the cell. Believe it or not, there's special proteins called motor proteins, and they can attach to these microtubules and literally, chemically, walk along the surface of the microtubule, and as a result, move materials within the cell. Let's take a look at what some of these things look like. There we have our microfilament composed of, as you can see, two chains of twisted actin subunits. Actin is the protein. The microfilament is a number of these twisted together, hooked together and twisted into a helical kind of chain. Intermediate filaments, you can see, are composed of bundles of proteins. So we're talking about proteins that are wrapped around proteins, creating these little fibrous subunits, and then those fibrous subunits are wrapped around each other until we get this intermediate filament. Notice we've increased in size from about 7 nanometers wide to about 10 nanometers wide. Finally, we have the microtubule, which is composed of the protein called tubulin, and tubulin comes in subunits that are made of two proteins. And if I want to make my microtubule longer, I add these proteins onto the tube structure. If I want to make the tube shorter, I pull these subunits off of the tube structure. Notice that we said that this cytoskeletal structure is something that can be modified and reworked. Notice the size difference, by the way, with the microtubules. Now we're at the largest protein structure that we find in the cytoskeleton. Microtubules about, what, 25 nanometers wide. All right, so the three components of the cytoskeleton, and a number of these are going to be mentioned again in terms of the jobs they have as we talk about other functions of the cell. For example, structures in eukaryotic cells called cilia and flagella are structures that extend out from the cell. They have a protein internal structure, microtubule protein internal structure, and they're surrounded by the plasma membrane. So don't think of them as sticking through the plasma membrane. Think of them as being the plasma membrane stretched very far out from the cell with an internal protein structure composed of microtubules. All right? So here's what we have. Notice the internal structure with the clusters of microtubules that are shown on the inside of this structure. By the way, if this were a flagellum, if this were a cilium, it would simply be smaller, but it would have the same internal structure. Flagella, a flagellum, is a larger structure that has basically the same internal organization. Notice that what the internal organization is clusters of microtubules. You can see that we have 9 pairs of microtubules making up the basic structure when we look at a cross-section. At the bottom of the flagellum, and this would also be true if we looked at the bottom of the cilia, we see a thing called a basal body, also composed of clusters of microtubules. Surrounding the whole thing, notice that there's a thin layer of cell membrane, right, surrounding all of this. So don't think of these proteins as sticking out naked through the surface of the cell. Instead, you want to think of them as being a very elaborate extension of the cell's surface, the plasma membrane. All right, now, the interesting thing about these structures is that the microtubules have motor proteins on the side of them. Remember I mentioned motor proteins are commonly associated with what? Microtubules. Motor proteins have the ability to change shape. I said that they walk along something. Really, the way to think of it is that as I give them energy, they change shape. They lose the energy, they change shape again, and the protein, by changing, having these two different shapes, without protein energy in the form of ATPs, we see the protein literally move, step along another protein structure. Well, here's a close-up of some of those microtubules that are found inside of cilia and flagella. Notice that the motor protein that I've been referring to here is given the name adenine arm. And so that's the motor protein that I've been talking about. Add energy to it. Notice energy is represented in the form of ATP energy. We'll discuss the idea of how ATPs add energy to molecules later. But ATP energy causes those little protein arms to reach across, grab the other microtubule doublet, and bend. Another one reaches across and grabs and bends. And as each one of these bend, the whole structure bends. The whole structure of the flagellum can then bend back and forth. Must happen very rapidly, huh? Back and forth as the flagellum whips and propels the cell. All right, so we're talking about a biochemical activity that's occurring very rapidly, but it's the result of proteins interacting with other proteins, gaining energy and losing energy, and changing shape as they do so. That's what provides the power to cause the flagella or the cilia to bend and move in their whip-like or wave-like motion. So now, what about other features of eukaryotic cells that are associated with support or structure? We talked about the cytoskeleton. Notice how the cytoskeleton was support, but also movement. Here we're talking about, what about where two cells come together, meet each other? Cell surfaces, cell junctions. Now, the cell surface in general, and I'm thinking about in particular things that are the plasma membrane or materials outside of it, are used to protect the cell. They might support the cell, and in many cases it might be used, materials outside the plasma membrane, as a way to hook cells together or join them with each other. So that surface of the cell is important. It's the way in which the cell is going to interact with other cells. It's also going to be the way in which the cell interacts with its surrounding aqueous environments. It's going to be a key determinant of what the cell's function, its job, its role is within a multicellular organism, or how it's going to function if it's a unicellular organism in its own particular environment. Now, if we look at plant cells, what we find is a nice example of this material that's external to, or just outside of, the cell membrane, and that's the cell wall of the plant cell, and it's composed primarily of cellulose. Now, through this cell wall structure, though, we might have penetrations. We might have connections that occur between one cell and the next cell, and sure enough, in many plants' tissues, we find that the cells do indeed have cell walls between them that help to join them to each other and provide that cell tissue, that plant tissue, with rigid support. But we find that those cell walls are perforated, lined with cell membrane, and the cytoplasm of one cell is in essence in direct contact with the cytoplasm of the adjacent cell. Materials can be moved through that passageway. The passageway is called a plasmodesmata, and you can see in this illustration the representation of that idea. Plant cells bunched up against each other with their cell walls touching, but at a number of locations, passageways, perforations through those cell wall structures, so that materials from one cell's cytoplasm can move to the other cell's cytoplasm. Excuse me. Eukaryotic cells and a number of prokaryotic cells have materials external to their cell membrane. You could think of the cell wall of the plant cell as being an example of materials that are extracellular, a type of extracellular matrix. And so we're going to see other examples of this. In a number of cases, organisms put sugars and proteins, and you probably say, well, wait a minute, Sean, you said that wall structures were basically different mixes of sugars and proteins. That's right. We're looking at variations on this basic idea that if I'm a cell and I want to have extracellular material, an extracellular matrix, the key way in which I do it is by making a mixture of carbohydrates and proteins, glycoproteins materials. Some cases it's sticky, some cases it's slippery, some cases it's very tough material. Now, that can be used to bind cells together in tissues. It might be used to bind prokaryotic cells together in what are called biofilms. So this extracellular matrix might be a type of glue, a type of mechanism to hold cells to each other. It also might provide the cell with certain types of protective or structural features. And so we're going to see in both prokaryotic and eukaryotic cells how the extracellular matrix helps define the nature of the organization that the cell is in. For example, the bony material in your bones is an extracellular matrix produced by the osteocytes, the bone cells within that bone. They've secreted the bony matrix. What is bone? Well, guess what? It's a mixture of carbohydrates and proteins that have had a lot of minerals added to that mixture. By the way, the same would be true for things like connective tissue as well. So think about that in terms of some of the examples of extracellular matrix found in your body. Cells, we said, might be joined together by the extracellular matrix, but we're going to find that in a number of cases, cells need a more robust connection. Maybe in some cases a more specialized connection. And so we're going to see that there are going to be protein connectors that are produced by some types of cells that hook one cell, the proteins in its cytoplasm and cell membrane, with another cell's proteins that are part of its cytoplasm and cell membrane. Notice the representation here of some of these different kinds of junctions: tight junctions, anchoring junctions, communicating junctions. And notice what they're showing in all cases. They're showing how the membrane of two cells, the membranes of two cells, are being fused or attached or interconnected in some way, utilizing proteins that are part of the cell membrane, and in many cases also part of the cytoskeleton of the cell. So we have anchoring junctions and tight junctions. Tight junctions, first of all, bind cells together and create, in a sense, almost a waterproof welded connection between the two cells. Materials would not be able to move down and in between these cells because of this tight junction. It forms a very good barrier to materials. So that's why it's said to be a leak-proof, right? A leak-proof junction. Anchoring junctions, which are also called desmosomes, are ways in which one cell can interconnect its cytoskeleton with another cell's cytoskeleton and create a tight junction between cells, a very robust junction between cells that helps them hold together. The intent is not to create any kind of leak-proof barrier, however. Communicating junctions, are also called gap junctions, are going to be specialized clusters of proteins that form passageways. Notice, here's the gap junction shown here, and here's one of those passageways where material from one cell can move through the gap junction or communicating junction to the other cell. Cells, for example, like the cells in your heart muscle, all right, those types of muscle cells, those cardiac muscle cells, have numerous communicating junctions, gap junctions. That means that chemical information from one cell can flow very rapidly to the adjacent cell. Very important for the fast responsiveness we want in that heart muscle tissue. All right, so these are some of the connections that occur between cells. Notice we're adding to the idea of how cells can be attached to each other. Might be through their wall, might be through some other type of extracellular matrix. Here we see that it might be through some kind of specialized protein attachment that occurs between cells. All right, so to summarize this discussion, you can see that we've sort of jumped around and have talked about a variety of different types of organelles. But first we focused on the chloroplast and mitochondria and their role in energy conversion. We'll talk about both of those in more detail later, and also we'll talk in more detail about the functions of energy conversion. We talked about the cytoskeleton, how it's associated with providing support structure for the cell internally, made of proteins. Remember, microfilaments, intermediate filaments, and those microtubules. The microtubules were directly associated also with, right, activities of movement. Movement within the cell, but also movement of cellular appendages called flagella and cilia that could move the entire cell. And then finally we talked about how material that's extracellular, that's outside of the cell membrane, an extracellular matrix, might be associated with forming connections between cells. We talked about that in terms of the cell wall of plants. We talked about that in terms of the extracellular matrix that might be found in your own body. And then we went further and said there's other forms of connections between cells that can result from the actions of proteins that are part of the cell membrane of the cell and may be interconnected to the cytoskeleton of the cell as well. So you can see, even though we've talked about a variety of different kinds of organelle structures, in many cases we've shown how they interconnect, how they interact. That should be your expectation. Now we'll have an opportunity to talk about some of these in much more detail later. But use this information to help you complete your table of cellular organelles and their structural and functional qualities. Thanks very much. I look forward to talking to you next time.